Parasites. Nathan's right, 100%. We all have it. The CDC says maybe they give such a broad range, anywhere from 20 to 80%. That's pretty bizarre. But the truth is you can't find it. These guys don't hang out in your bowels ready to be discovered. The reason they're called parasites is because they successfully avoid being discovered and killed. All right, so they'll burrow in your bowel wall, your liver, your lung, your brain, your ovaries, your testes, anywhere they can, and they'll have up to 2,000 eggs per day. Okay, so now I want to go through some stuff here. So let's see. This is kind of amazing. There's about 1 billion different species of microbes. 1 billion different species. Okay, and we haven't discovered 99.999%. They came up with this number. I'm not sure if it's a real number, but they call it a nonillion, which is 1 to the 30th power, is the estimated number of microbes here. But the microbes we find them all the way down 2 kilometers in the ocean and 77 kilometers up into the atmosphere. There's only one place on Earth where we can't find them, and that's this well in Ethiopia where it's high saline and high sulfur, boiling. But that's the only place on the planet. They're also everywhere in our body. There's no place that they're not. They are responsible for recycling sulfur, recycling carbon, without them, there is nothing. And in fact, they are the context in which us macroorganisms exist. Without them, we don't exist. So this whole thing about germs and all that is madness. You understand that? You know why you get the colostrum from a cow? Because the cow's face is in the dirt all day. It's got a strong immune system. So you don't stay clean in your middle-class house, get dirty. Because guess what? Guess what's going to happen when you breathe your last breath? You turn back into Earth. Not Ethiopia, not America, not China. Earth. Earthlings. Okay, so anyway, we have to eat more dirt. Yeah, so here, this is another fantastic thing. 60,000 different species in just, 10 centimeters of soil. It's amazing. Okay. Now, its relationship to cancer, and this is very important. So back in, 2012, 17.8% of the cancers that they discovered, I'm sorry I have to use that word, I hate that word, it's a Rockefeller word, I prefer CFCs, chronically fermenting cells, because that's exactly what they are. I always thought cancer was somewhere between Gemini and Leo, but that's not what the Rockefellers think. Okay, so anyway, so 17.8%. So what's going to happen by, they're predicting that by 2050, most CFCs will be from infections. Will be from some sort of microorganism. So if you look at the details here, if you look at the details here, you'll see the different contribution of the different kinds of parasites and other organisms. So they look at funguses, viruses, if they exist. I'm not sure they do, and because they're not biological. They don't eat, they don't drink, they don't breathe, they don't procreate, they have no waste, they have no metabolism. And you can make them in Bethesda, Maryland. And then you can export them to Africa and to the gay community. That's what they do. Where are we? Okay, here we go. So just our relationship with microorganisms can be that mutualism, both species are benefited, commensalism, where one species is benefited, the other one is unharmed, and then parasitism, where both are, where the one is benefited and the other one is harmed. Okay, so we have a lot of commensalism going on in our gut. Most of our gut is commensalism, right? Meaning we're benefited, well, they're benefited, and we are too. Actually, it should be considered mutualism because without the microorganisms in our gut, we wouldn't be able to live. Just like if we didn't have a heart, we didn't have a liver, we didn't have kidneys. If we didn't have them, we wouldn't be able to live. So therefore, are they really them or are they a part of us? Anyway, this is what a parasite is. all these different. We're going to focus on the protozoa and the helminths, which are worms, okay? And that's what we usually think of when we say parasites. But a protozoa is a single-celled organism, and a helminth is a worm, okay? And I just wanted to show you this because it's called molecular pathological epidemiology. And basically, just look at that middle slide with the underlines. Every gastrointestinal CFC has associated with it a specific dysbiosis. You all know what dysbiosis is? When you have it's not, the proportions of the microorganisms in your gut are not in your best interest. The proportions are out of balance. All right, so people with esophageal CFCs will have one kind, people with colon CFCs, liver, and even breast. We're now finding breast too. So the implication is that in the future, we might be able to just, even maybe future's tomorrow, just do a fecal transplant of a woman who doesn't have breast CFCs into that woman. And let's see what happens. Anyway, the mechanisms of survival, these guys, are immune evasion and then non. I'm not going to get into the details of that. Here's the worms. There's, the nematodes are the roundworms, and they're the most prevalent in us. And then there's the flatworms, which are flukes and tapeworms. When they estimate 2 billion people are infected with at least one of the main four, again, the truth is they're everywhere, they're all the time. We're host to 60 species. So that's where they live. So you have them, we have them. That has nothing to do with whether or not you eat corpses or not. A lot of people eat corpses still, I know. I think in 100 years from now, there's going to be corpse eaters will be, well, did we really do that? Yeah. But anyway, I thought it was only on Halloween, right? Isn't that what a ghoul is? One who devours dead bodies? Yeah, remember that, on October 31st. Anyway, but even people that are vegan, because these roundworms, a lot of them have a second part of their life cycle in the soil. They have to get into the soil. So you could get them from just eating a carrot. So it's not just the corpse eaters, it's everyone. Okay. So now I wanted to point this out. Okay, the most common causes of CFC death is lung, liver, stomach, colorectal, breast, and esophageal. 4 of those come from microorganisms. And we won't go into the details of them all because we got to get through this. Now, you've heard of schistosomas, right? Maybe? Okay. Anyway, there's 3 different species we're going to look at. These are they're like the ones I told you that have to have a part of their life cycle in the soil. Well, these guys, they come out in the feces, they somehow wind up in a fresh water, they go into a snail, and then the snail winds up, it transforms into another form, and it goes into a fish, and then the people eat the fish. So it's got 2 intermediate hosts. Usually in tropical and subtropical, but they're finding it in Appalachia now. They're finding it in the southern parts of the U.S. And some of the parasites that they used to find only like in the Amazon River are now in the Mississippi River. Okay, so it's just the way it is, But anyway, the schistosoma, they say 240 million people in 76 countries. So anyway, schistosomas, these are what they call flukes, but so is Clinorchus and Opistorchis. The reason that the Opistorchis is something that I know pretty intimately is because in Thailand, they're in Thailand, and in Thailand there's an area in Thailand called Isan, which is northeast, and they eat something called bala. They have the weird words, but they say bala, and then what it is, it's raw fish, but the fish that live in the rice paddies, and they eat that raw, and they get into their gallbladders, and they wind up getting, and it's got the highest rate of cholangiocarcinoma in the world is in Thailand, in Isan. So you know what they think? This is really great reasoning. This kind of reminds me of the reasoning that you might hear from someone like, what's his name? Anthony Fauci, that's right. They say that, well, you know what? If I got those in my liver, I'm going to just drink a lot of alcohol to kill them. They do. But anyway, so yeah, so these are the flukes. Flukes are little flatworms, and they're they're microscopic. You'll never see them. You'll never find them on a test ever. You'll never know it that way. Okay, but what I want you to see is what they do is they change the signaling pathways. The p53. You might know that people with CFCs often have down-regulated p53 genes. By the way, they're not really mutations, right? Because a mutation doesn't go away. You can upregulate a p53. You can upregulate a PTEN, and it's not a mutation. If someone has Down syndrome, it doesn't matter what you feed them, they're still going to have Down syndrome. That's a mutation. Okay, but they use this word because they want you to feel helpless, so you can go to them, and they're going to be on their white horse with their white coats. Doctor, it's madness. I'm telling you, these guys are sorcerers. Don't believe a word they say, not one word. You can tell when they're lying because they're awake. Yeah, but here's the thing. It's interesting. It's not just the adult worm. It can be their eggs, right? Their soluble egg proteins. And it's a lot of times it is their eggs that are challenging the immune system and causing problems. And I'm not going to get into the details because, yeah. So Mansoni not only because the other there's three basic species of the schistosomas that we look at, but Mansoni is with prostate and colon. And one of the big things they do is in our immune system, it can be polarized towards what's called Th1 or Th2. Th1 is when your immune system is on it. It's going to kill whatever's there. Th2 is chronic. And when you get into a Th2, then you're going to actually support tumor growth. Okay, so you don't want to be in Th2. You want to be in Th1. Well, these guys put you into Th2. Okay, but it's the schistosoma hematobium that is the one that's associated with bladder CFCs. Now, most bladder CFCs that people would get are urothelial. That just means that the actual cellular structure is what is called urothelial. But the ones from schistosoma are squamous cell. So that's how you know the difference. Because we had a missionary at our clinic who had been down into these areas in Africa, and she had bladder CFC, so I thought for sure it's going to be schistosoma, but it wasn't. It was urothelial, not squamous cell, so we know it wasn't from the parasite. Again here's the Opistorchis. And as I said, the main way that Opistorchis works, and this is not just for that, any of these guys, is it upregulates IL-6, which is a highly inflammatory interleukin, okay, and that sets the whole thing in motion. By the Well, no. There is a parasite which we'll talk about in a minute that will crawl through your feet, like while you're walking. They'll crawl on your feet. They get into your capillaries. They go up in the blood to your lung, and then they settle in the walls of your lung. They produce eggs. And then the eggs produce these little like tiny worms, filarial. They come up into your throat, and you go, you swallow it, and then it goes down into your duodenum. They lay more eggs, and it comes out in your feces. And these guys are really clever because sometimes when they come out in your feces, they crawl right back into your. They find another area on your. Yeah, so they're a lot of fun. So, yeah. So I mean, I'm on my fourth round of a parasite cleanse. Because they're there. Okay, now. Those were the worms, okay, for the most part. Now we'll talk about the protozoa. The protozoa, what I want to bring to your attention, you may have heard of trichomonas, right? Women get that. Men are silent carriers. So in other words, they don't know it, or they're keeping it a secret. Probably the latter, knowing men, right? So, but women know it because they have an odor like fish, so it becomes pretty clear, right? And you can take something like metronidazole or nitidazole. What is it again? Nitazole? Tinidazole, yeah. And they work really well for the protozoals, the antiprotozoals. So does something called Alinea. Alinea works for the antiprotozoals. The other ones, even though ivermectin, like, is an anti-helminth, an anti-worm, it can get some of the protozoas. So what we have to do is we have to take multiple, 3 weeks on, 1 week off, 3 weeks on, 1 week off, and do 5, 6, 8, 9, 10. And I'm going to show you a couple cases at the end here, if I can get through it. Do I have time? How much time do I have? Okay, so, but anyway, so Trichomonas is well known. The thing about Trichomonas, if you don't recognize it and you don't treat it, it can, and you're a woman, you can get cervical CFCs, and a man can get prostate CFCs. Okay, so they're not just that, they're not just benign. You know, when we think of STDs, we think of them just having some local effect, but they can have long-term effects as well. Another protozoal, of course, is malaria. You've all heard of malaria. Anyway, and then let's see. Oh, so the way these guys work, when they get into the cell, of course, how are you going to find these protozoa? They live inside your cells, so you're not going to find them. But they get inside your cells, and they get in there, and they get in this little vacuole. They use the membrane, and they make it so that it's like a filter, and it filters in all their nutrient requirements, as well as the material needed to make DNA and RNA. So they get everything they need in these little vacuoles, and they're protected. You know, remember, they're called parasites, so they're good at this. They've been doing it a long time. Yeah, and just a thing here about malaria. What it does, actually, is it inhibits the Epstein-Barr specific T cell immunity. In other words, it protects Epstein-Barr, and then Epstein-Barr will result in Burkitt's lymphoma. And we know that Epstein-Barr not only does that, Epstein-Barr has been associated with nasopharyngeal CFCs, and they used to call it the kissing disease. Everybody, by the time they're 18, 19 years old, is positive for Epstein-Barr. It used to be called mononucleosis, and things like that. Could CFCs, cancer, actually be a parasitic infection? And this has been debated until today. Is what we're seeing really a parasitic infection? And so all the way back to 1850, I think was 1860 is when they started talking about it. But these guys here, von Hoekelum and Hauser, 1890, they found evidence. Metchnikov, the director of the Institute of Pasteur, found evidence. What they're finding is these little organisms that are in malignant cells but not in healthy cells, right? But in those days, they didn't have any way of dealing with them. And then finally, there was a guy named Roux, I don't know how you say it, R-O-U-S, but he's French. I don't know how you say it, but whatever. He was able to actually infect and cause CFCs. They call it the Roux sarcoma virus. Peyton Roux. Now, probably one of the biggest issues in all of parasitology and microbiology, et cetera, is the concept of polymorphism versus monomorphism. So monomorphism says that organisms have one shape, one form, and that's it. Polymorphism says no. They have through their life cycle, they have different things. Well, that's obvious. I mean, check out a caterpillar. Becomes a butterfly. I mean, so that they're not even microorganisms. But anyway, that's been the argument, right? And polymorphism says also that that points the finger at the milieu. So they have found, and these guys, I'll show you in a minute, these guys have found that in one environment, it's a fungus. In another environment, it's a bacteria. In another environment, it's a mycoplasm. They have evidence. I'm not just making it up. So pleomorphism is a real thing. And think about it. If our context of macroorganisms is because we have microorganisms in and around us, and if they will change and become what is necessary to maintain this thing called life. So it's really not a far-fetched concept. This is right in the perfect place so that I can't see it with or without the glasses.
Whoever did that was great. Right. So yeah. So basically, they could not be detected in people that didn't have the condition. Okay. And the big one. Yeah. Let me see. Yeah. This is a point I wanted to make really, really good here. Was this woman? I can't pronounce her name. Lebedewa. Anyway, that was her. I guess her pseudonym. I would have picked a much easier pseudonym. Anyway, she was a very respected researcher. She concluded that all CFCs were due to trichomonads, like we talked about trichomonas. But what she found was they become different forms, so they become cysts. Now, what I've come to believe is, what's a cyst? Like you do an ultrasound, you go, and the radiologist says, oh, you got a couple cysts on your kidney, and you got a cyst in your liver. Okay, well, what is a cyst? We don't really know what a cyst is. Now I know. I think they're probably parasites, and that's how they're hiding out. Okay? We don't worry about that. Yeah, let's do a mammogram. Sure. Let's squash your breasts and then irradiate them because it's good. Yeah. That makes a lot of sense. You notice they don't do that to the testicles, right? Just to the breast. I wonder why do you think that is? Oh, men invented it. That was it. Yeah. So. Okay, so now I want to talk about Dr. Weber. Dr. Winter and Scheer, and then we'll talk about Weber. At the Max von Pettenkofer Institute in Munich, 1970s, they were doing this work. Okay, so this is not 100 years ago. This is 1970s, 60s, and 70s. But anyway, the guys I just mentioned found the same thing that Alphonse Weber did. What they found is that these parasites and the CFCs, they have the same survival mechanism. They produce the same substances and metabolites. They reprogram the immune system, and they're killed by the same drugs. Fenbendazole kills them, and it helps out Joe Tippins. It's great. Who would have thought? So the question is, are parasites and CFCs, how are they related? They're obviously related, right? Anyway, so this is what he proved, and I'm going to show you this little. I hope it works. I have these little films in here, but basically, what Weber found was these little organisms that would go into the red blood cell. They would eat the hemoglobin, and then they would grow, and then they would burst out, and then they would go into other ones. Eventually, they got eaten by a macrophage who took them to the liver, and they exploded in the liver. I mean, you wind up with CFCs.